what are the ideas for which is maybe most significant here, and any lessons learned as we think about future programs developing adeno-based vaccines that could be mucosally directed?
Who would you like to go first?
It could be any of you. Dr. Douglas, why don't you start, since you were just presenting?
I think first lesson learned is I think all vaccine developers are cautious in the interpretation of preclinical data, but it's clearly particularly difficult to optimize administration and infer clinical performance from preclinical with these intranasal vaccines. I think also our adenovirus serotype wasn't selected for transduction of respiratory epithelium. And I think there is some work to be done in choosing and seeing whether there are real differences between serotypes and their efficiency of transduction of mucosa.
Thank you. Dr. Allo, what are your thoughts?
Yeah, I think just to add on to Dr. Douglas's findings, no doubt there are a few differences amongst the three vaccines, the CHAD or the vector-based vaccines that we spoke about today. I think it was really important for us to get the formulation right on this vaccine candidate. One could also argue that a 0.5 ml dose presentation is quite a lot for the nose, but I think there is a certain amount of postnasal drip that happens, and if that happens into the windpipe, I think that's a beneficial impact for the participant. Of course, with the formulation, this is, I think, as soon as we licensed this candidate and embarked upon this project, that was probably the first thing we were thinking about. How do we get this formulation right? How do you make sure it's immunogenic? Dr. Douglas nailed it by saying preclinical data is just not enough. Sometimes animals do lie, and we're fortunate to be very cautious with our phase 1 and phase 2 studies. We did take some amount of time getting some important results from that, doing a decent amount of dose ranging. We did move from 0.2 ml to 0.5 ml in our nasal vaccine program. So I think it's a combination of things with formulation, increasing the dose volume. Perhaps the transfection of the adenoviral vector for us is slightly better than the AZD vaccine. And also seroresponse or seronegativity could also play a factor. But I think it's worked out well for us, and I think we're moving on to trying to figure out how best this curbs infection and transmission.
Thank you. Dr. Zhu?
Initially, actually, we didn't expect that inhalation could give so strong humoral response. And the initial shooting is for IgA and mucosal immunity. But we found out that this vaccine, by inhalation, can induce so strong IgG. Then we go back to think about what is the feature of this adeno5. I think one thing maybe is because the adenotype 5 actually is it infects respiratory tract basically, and it's targeting for macrophage and other like dendritic cell in respiratory tract, like in lung, and that may be the one reason that it can induce very strong immune response. That's the one reason, and we have done twice in monkey to test whether if the intranasal works better or in comparison with the inhaled version. And that data actually support us to go for inhalation.
Thank you. And Dr. Tucker, I know you also have a adeno-based product. So I'd be interested to hear your thoughts in terms of why you selected Ad5 versus other potential vectors and other compounds.
Yeah, well, why we selected Ad5 because it worked in humans as an oral tablet. So that was, and it's a lot of experience. So I just wanted to go back and really compliment, Dr. Douglas on, when the data that he showed, I mean, basically what he did is he did the right experiment. He looked for the nasal IgA responses, and yes, it didn't work, but it was very clear that, that he was doing the right thing. And I just, again, I want to point out that, when we're starting to look at nasal IgA responses, it's important to kind of look at it in the same manner. The other thing, of course, is I would like to point out, of course, that the best nasal IgA response in humans has come from the oral tablet vaccine. Just to be clear, I don't think there's been anything that's been better presented today, for sure, that I've seen in the literature than that norovirus study that I presented in humans. So, I mean, I think that we keep thinking that, nasal response has to be elicited by a nasal vaccine. And I think that, CanSino aerosol looked really good. And I think our vaccine data looked fantastic. So,
Dr. Ella, you had your hand up?
Yeah, sure. I just also wanted to address the concerns on using intranasal vaccines for pre-existing immunity. We had conducted 2 studies, and the second study was a heterologous booster study where we had enrolled participants that had already been previously vaccinated with Covishield, which is the vaccine developed by the Jenner Institute. And because both these vaccine candidates are based on Chad vectors, what we did notice is that there was no cross-neutralization between strains. Though they're different serotypes, there was no cross-neutralization. And then we came to the hypothesis that our vaccine is able to bypass any pre-existing immunity generated by the AstraZeneca vaccine. Can we give our vaccine as the second booster, third booster? Now that's a question I'll have to come back to you upon. But one of the biggest doubts that we had in our mind was, can you give this as a booster to participants who received the ChAdOx vaccine? And the answer is yes, and it is bypassing immunity.
Great. And before I pass things over to Dan to ask some additional questions, I wanted one more question on the adeno-based work. There's been some belief that, or hypothesis that if you deliver an adeno-based vaccine mucosally rather than by injection, that you might be able to avoid the thrombosis side effects that have been observed. And of course, it's so rare, it's very hard to design an experiment on that, but I'm curious your thoughts on this, on the benefits of mucosal delivery from that safety perspective.
Well, we think that's true because you're not putting it into a area of, the veins or anything that could potentially lead to that. But we haven't studied 1 million people, so I can't answer that definitively. So
I'm a believer in that theory as well. I think the data that interaction of the vector capsid with platelet factor 4 is a prerequisite for the development of this very, very rare condition, to me personally is relatively convincing and would not be expected to occur after mucosal administration. The tricky bit, of course, is going to be persuading regulators in particular about that negative change. And I think we need better preclinical models of this condition, which is actually quite tricky to show that change in route does have that effect.
Great, thank you. Dan, I'll pass the next set of questions to you.
Great, thanks. And speaking of preclinical models, so maybe go around the room here, but in terms of any research tools that haven't been developed yet, are in development now, that could be useful, whether they're animal models, immunoassays, et cetera, that could have either benefited the selection of your candidates had they been in place, or have benefited the selection of your candidates because they were in place. Maybe just go around the room. Can you all kind of speak to maybe some of the biggest impact, whether it's animal models or assays, that could be useful for pushing some of these mucosal vaccines further down the pipe?
I guess I could start this, Michael Egan at Castlevax. Having been in the vaccine research and development field for more than 20 years, I've kind of evolved into a fan of not making huge investments or overdoing the preclinical work. Get enough preclinical work to demonstrate that you've got, proof of concept. And then knowing that sometimes, animals lie, mice, monkeys exaggerate, get into the clinic as quickly as possible and measure the types of things that you feel are important, in the clinical situation. With regard to other tools that would help us, I think, in our clinical development efforts is, I think, better assays for mucosal immune responses, better collection and sample processing procedures, and maybe some really robust assays that are available across the field. So we're all kind of, maybe working with one or two partners and who are doing kind of the same sort of sample collection, sample processing, and then a mucosal immune assay. So we can have, maybe apples to apples comparisons at the end of the day.
Anybody else want to jump in?
Well, I'll just echo. I think Dr. Egan's right on that. It's really important to basically get the humans fast. And I made the argument that, of course, with SARS-CoV-2, we've been pounded by coronaviruses in our entire lives, and it's going to be awful hard to replicate that, when you're talking about an animal model. So that would be my comment on the preclinical systems section.
David,
I think.
Sorry, go ahead.
No, I think one point that we realize is that, when we are doing hamsters, right? Hamsters is like the gold standard now for all these respiratory infections. But there are not enough tools, for example. There are no IgA reagents, when you're dealing with hamsters to really look at how deep you can understand the mucosal response. That's number one. Number two, when it comes to, again, to look at, let's say if we are getting the blood from our phase 1, how do we compare and standardize, because we are kind of going in an environment that's constantly changing. And if you're bringing a new technology into this phase 1 studies, these tools is, something that has to be somehow standardized, I guess, because you're not in a naive settings anymore, but you are looking at a different thing. Obviously, you look at the baseline, but how do you measure, serum responses against the nasal responses, and then the mucosal responses? So that's what I think that first, if we have a very good animal model, obviously, this is what we always been told when we're looking at veterinary. Don't do too much mice experiments. Go to target animals as soon as possible. See the animals work or not. If it's protected, you have a vaccine. If not, just don't do anything else. But when it comes to here, you don't have the challenge model in humans. Although there are possibilities that you can actually establish a challenge model in humans, but until then, how would you really make sure that what you're seeing is real with the vaccine?
We heard yesterday from Chris Chu about the challenge model, and I think this is a very, very good use case for controlled human infection.
For full disclosure, we've announced that we're going to do a human challenge study on our COVID vaccine candidate. So that would be one of the next steps, because again, from our standpoint, being in this mucosal business for years, the correlates could be different. And, if you basically looked at the serum for how you respond, it may not tell you the full story about protection. And that was our lesson from the flu challenge study sponsored by BARDA.
Yeah, I would just like to echo that point. I think, as vaccine developers and, NIAID, CEPI, as the scientific community and funding organizations, I think we're all collectively failing. Dr. Zhu and I, and of course my colleagues on this panel have also described some great data, very promising data. We have 2 vaccines that are licensed in the world, and I think it's time that we all work together and try to figure out what's the best way to clinically establish these vaccines from an infection and transmission model. And I'm pretty sure you'll have at least the manufacturer support.
And do you have any other things you want to bring up? We've got just about a minute left.
With just a minute left, I think we've hit kind of the most important points, but no, any other questions for you, Rebecca?
Well, I guess I'd be curious to just hear if anyone has thoughts about kind of the ideal regimen. Should we be pursuing heterologous prime boosts in terms of combinations of intramuscular and mucosal driven But what are your thoughts on the best way to approach this? Is it too much to try to expect the ideal systemic and a mucosal response in a single product? Dr. Tucker.
Well, it's probably too late now to basically start over, because everybody's been hit with so many different vaccines and infections. So I guess you're just going to have to deal with the heterologous, unless you want to go to the youngest of kids and start, with mucosal, because you want to really elicit those cross-reactive responses. You're just going to be stuck with, all comers and backgrounds with all different vaccines and all different types of infections. So.
Yeah, one thing that we're doing at Castlevax in our phase 1 study and may continue to do in our phase 2 study is we're evaluating intranasal delivery by itself as a booster vaccine, and then also concomitant intranasal and intramuscular administration. In other words, the subject gets an IN dose of vaccine and an IM dose of vaccine at the same clinic visit. And we're asking the question, what's the immunization route that gives us, first of all, robust mucosal immunity and maybe even balanced, boosters of the systemic immunity. And so it'll be interesting to see, what those results look like, and then maybe what's the appetite or the acceptability of a vaccine that you have to sit down and get by 2 different routes during a single clinic visit. And if that's what it takes to get the robust and balanced, systemic and mucosal immunity, it'll be interesting to see if that's something that people feel is, can be developed and delivered commercially.
Yeah, very interesting. Well, I see we're out of time. I want to thank all of our speakers and panelists today. It was an excellent set of presentations, and really appreciated your insights during these discussions.
Thank you.
Yeah, thank you all. Really great. I learned a tremendous amount from these discussions today.